Good day, everyone. We are Group 5 and our topic is about protein engineering. Protein engineering is a fairly new field of study in both the engineering and in the biotechnology field. It involves the synthesis of new proteins. It does revisions in the current existing protein sequences or structures, and it is mainly performed to achieve desirable functions. In this research, or rather report, we will be dealing with the people who are directly involved by the use of protein engineering and also those who have benefited from it. We will also be talking about where its application is and its status here in the Philippines. Before we dive deeper into protein engineering, let us first show you a short video regarding our topic to help you visualize what basically the study revolves around in.
Proteins. They are the nanomachines that make biology happen. Every thought you have, every action you take, begins with proteins doing the job they were made for. To keep the flame of life burning, your body makes hundreds of trillions of proteins every second. But until fairly recently, scientists struggled to make even simple proteins. Biotechnology changed that. It cracked the genetic code that runs the protein-making machinery of the cell and ushered in a new era of protein-based medicines. Proteins that help restore essential functions impaired by disease. Proteins that strike disease with unmatched precision. It takes a rare blend of imagination and talent to engineer new types of proteins. And some of the world's most creative protein designers, they work at Amgen. Their expertise allows us to focus first on the biology of disease, confident that we can create the right tool for the right target. Our scientists have given Amgen the industry's largest collection of drug modalities. Novel tools like fusion proteins, bispecifics, and engineered viruses. Innovations that show the power of biotechnology to accelerate medical progress. Biology is astonishingly complex, and so is disease. That's why patients need new treatments that are sophisticated and varied, like nature itself. By increasing the versatility of medicine, Amgen scientists are bringing new options and hope to patients facing serious illness.
The people behind or involved in protein engineering. When the first protein was rationally designed during the 1970s and the 1980s, the sequence for this was optimized manually based on analysis of other known proteins. The sequence composition, amino acid charges, and the geometry of the desired structure. A professor from RWTH Aiken University reported the first chemical synthesis of an enzyme. The name of this professor was Bernd Gut, where in 1969 he and his team reported the first chemical synthesis of an enzyme. Since that time, his research interests have focused on the designs of novel functional peptides and proteins for basic research as well as biotechnical and medical applications. For example, dioxin, DDT, and HIV enhancers binding peptides, and for the isolation of recombinant human interferon. The next person will be Robert Bruce Merrifield, born July 15, 1921, and died in May 14, 2006. Was an American biochemist who won the Nobel Prize in Chemistry in 1984 for the invention of solid phase peptide synthesis. He is one of the members on Good Theme that reported the first chemical synthesis of an enzyme. His laboratory is also the first one to synthesize bradykinin, angiotensin, desaminoxytocin, and insulin. The next one is Ralph Franz Hirschman, from May 6, 1922 to June 20, 2009, was a German-American chemist who led the team that was responsible for the first organic synthesis of an enzyme, ribonuclease, exactly the same as burnt goat but using a different method. The comparatively simple 124 amino acid structure of ribonuclease made it a logical target for the first enzyme to synthesize. Hirschman's team built the enzyme in amino acid groups from 6 to 17 in length, which were assembled into large sections that were linked together, while Merrifield's approach was to assemble the entire enzyme by linking one amino acid at a time at the end of a chain. The next one would be David Baker, born October 6, 1962 in Seattle, Washington, is an American biochemist and computational biologist who was pioneered methods to predict and design the three-dimensional structures of proteins. Baker's group developed the Rosetta algorithm for ab initio protein structures prediction, which has been extended to a distributed computing project called Rosetta at Home and Foldit. The project aims to produce structural models of protein complexes as well as individual polypeptide chains. People who benefited from protein engineering. Protein engineering has a very important role in the food industry. Protein engineering of enzyme is a faster, more controlled, more targeted, and more accurate method to optimize the properties of enzyme for a specific industrial application than the traditional method.
Tackling some of the application of protein engineering, which are too many today, we have listed some of them, mainly food industry, environmental applications, medical applications, and biopolymer production. Important application area of protein engineering regarding food industry is the with gluten proteins. Their heterologous expression and protein engineering has been studied using a variety of expression systems such as E. coli, yeast, or cultured insect cells. Wild type and mutant with gluten proteins were produced to compare them to each other for protein structure function studies because of their availability, rapid and easy use, as well as high expression levels. Food industry makes use of variety food processing enzymes such as amylases and lipases, the properties of which are improved using recombinant DNA technology and protein engineering. Environmental applications of enzyme and protein engineering are also another important field of protein engineering. Genetic methods and strategies for designing microorganisms to eliminate environmental pollutants and included gene expression regulation to provide high catalytic activity under environmental stress conditions, such as presence of toxic compound, rational changes introduced in regulatory proteins that control catabolic activities, creation of new metabolic routes and combinations. The use of protein engineering for medical applications for cancer treatment studies is a major area of interest. Pre-targeted radioimmunotherapy has been discussed as a potential cancer treatment. By pre-targeting, radiation toxicity is minimized by separating the rapidly cleared radionuclide and the long circulating antibody. For pharmacokinetic properties of antibodies have been improved by protein engineering and antibody variants of different size and antigen binding sites produced for the ultimate use as imaging probes specific to target tissues. Protein engineering applications for biopolymer production are also promising. Particularly, peptides are becoming increasingly important as biomaterials because of their specific physical, chemical, and biological properties. Protein engineering and macromolecular self-assembly are utilized to produce these peptide-based biomaterials such as elastin-like polypeptides and silk-like polymers.
Protein engineering is not yet well known here in the Philippines, but despite of that, there are some articles that can be found about protein engineering in the Philippines. Upon researching, there are some studies about protein engineering that was conducted here in the Philippines. One of them is entitled Protein Engineering of Mung Bean. In this study, it shows the food protein engineering improves the nutritional value of common food crops such as mung bean. Having successfully mutated the lactostatin peptide into the AS-globulin, this product can be further developed into a food supplement or additive for use by individuals suffering from high cholesterol levels. Last 2018, University of the Philippines hosted a protein engineering workshop for free to increase the technical proficiency of our young scientists and researchers. It is envisioned to improve the skills and knowledge of researchers in current methodologies in protein research.